Hello and welcome to today's session. This session focuses on learning outcome to LO2. Understand the data collected and draw appropriate conclusions. In this video, we will explore how data interpretation can enhance your biology experiments. You will learn how to analyze collected data, draw meaningful conclusions, and effectively link your results back to your hypothesis. Interpreting results involves using tables, calculations, or graphs. Identifying patterns and trends is crucial. Use biological knowledge to explain outcomes. Draw conclusions supported by evidence. Determine if the hypothesis is supported. Compare results under different conditions. Refer to numerical values for accuracy. Identify increases, decreases, or stability. Recognize anomalies in your data. Use mean values or graphs for clarity. Conclusions should align with the aim and hypothesis. Base them on collected results, not assumptions. Use correct biological terminology. Avoid introducing new information. Clearly state what the results show. In osmosis experiments, low concentration solutions cause mass gain in potatoes. Higher concentrations lead to mass loss. Water moves by osmosis through cell membranes. Results support the hypothesis about solution concentration. This demonstrates water potential differences. Higher enzyme concentration reduces reaction time. More enzyme molecules form complexes faster. This increases the reaction rate. Results support the hypothesis. Enzyme concentration directly affects reaction speed. Choose the right method to present data clearly. Use tables, calculated values, or graphs. Match the method to the data type. For osmosis, use a results table and bar chart. This ensures clarity in trends. Graphs illustrate relationships between variables. Use bar charts or line graphs for clarity. Label axes appropriately for understanding. A graph shows trends clearly. It's an essential tool for data interpretation. Describe clear trends in the data. Use specific values, not vague statements. Identify any anomalous results. Explain why a value is anomalous. Focus on what the data shows. Potato chips lose mass as squash concentration increases. 0% concentration shows mass gain, 10% shows minimal change. Higher concentrations cause greater mass loss. This negative correlation is clear. Increased enzyme concentration shortens reaction time. 1% concentration takes 46 seconds. 2% reduces it to 32 seconds. 3% further lowers it to 20 seconds. This shows a strong negative relationship. Confirm trends with consistency across data. Consistent patterns indicate reliability. Minor anomalies may occur. They should not disrupt the overall trend. Reliable data supports strong conclusions. Mass loss increases with squash concentration. Lower concentrations show mass gain. Higher concentrations show mass loss. This pattern is consistent and repeatable. Anomalies are minor and explainable. Higher enzyme concentrations consistently reduce reaction time. The trend is strong across all repeats. One anomaly at 2% is minor. It doesn't disrupt the overall pattern. Consistency confirms the relationship. Consult key resources for in-depth understanding. Bell's Guide helps in research projects. Ditham's book covers statistics for biologists. Kirkup provides insights into experimental methods. Peacock's work focuses on teaching biology. Refer to ebooks and journals on Moodle. For queries, email the provided address. Submit assignments after unit completion. Follow the submission guidelines carefully. Ensure all tasks are completed on time. In conclusion, the video effectively highlights the key concepts and findings discussed, providing a coherent synthesis of the topic. The arguments presented are well supported by credible evidence, demonstrating clear connections between theory and practice. Overall, the content offers reliable insights, reinforcing the importance of the subject matter and its practical implications. You can reach us on WhatsApp at 0748-436-1688 for any queries. A team member will be happy to help. Thank you, and see you at the following session.